So the domain 5 is reproducibility and replicability of methods and results in the same model. Now this is different from domain 4, which looked at the clinically relevant variations. Here it is about the same model. Now, a little bit of background before we go into the purpose of this domain. In a survey of more than 1,500 scientists conducted by Nature, more than 70% of researchers tried and failed to reproduce another scientist's experiments. More than 50% failed to reproduce their own experiments. About 90% of scientists surveyed thought there was a slight or significant reproducibility crisis. So, the purpose of this particular domain is to assess reproducibility and replicability of the methods and results in the same animal model. This differs from external validity, which focuses on whether the results were reproduced in a different clinically relevant model. Now, a little bit of explanation about some terms. Reproducibility is the ability to achieve similar or nearly identical results using comparable materials and methodologies. So it's similar. Here the word is similar, or comparable materials or comparable methodologies. Replicability is the ability to repeat a prior result using the same source materials and methodologies. So there is a subtle difference between reproducibility and replicability. There is also another term that we will be using, what we call as protocol. Protocol in the scientific context is the steps or methods followed in the scientific experiment. We do not mean anything else in this particular context. We mean steps or methods followed in the scientific experiments. That's what we mean by protocol. The first signaling question: did the authors describe the experimental protocols sufficiently to allow their replication? One of the methods of improving replication is to describe the experimental protocols sufficiently. Classify this as yes or probably yes if the authors have described the experimental protocols sufficiently to allow their replication. Whether the experimental protocols are described sufficiently is a subjective assessment. Classify this as no information if it was not clear whether the experimental protocols were described sufficiently to allow their replication. Classify this as no or probably no if it is clear that the authors described the experimental protocols insufficiently, so it's not possible to replicate the findings using the details provided. They have provided simply insufficient information. Next signaling question, 5.2. Did an independent group of researchers replicate the experimental protocols? Now this signaling question is different from 5.1. The previous question assesses whether the protocols were described sufficiently, while this signaling question assesses whether these protocols were actually replicated by an independent group of researchers. Okay, so this is more objective. The previous one was very subjective. This particular signaling question is more objective because following that protocol, were they simply able to carry out that protocol using that information. The independent group of researchers, here we are considering a very broad group of people. It could be part of the author team, or it could be different from the author team. It could be from the same institution or different institution. The key here is they should repeat the experiments independently. So they should be able to carry out the experiment independently. That's what this signaling question is about. Previous signaling question was about was it described sufficiently, which was very subjective. Here we are asking, did they actually do it? So classify this as yes or probably yes if these protocols were actually replicated by an independent group of researchers. It could be based on a literature search, so you could replicate somebody else's experiments. or it could be from the same study where you have an independent group of researchers uh, from the same institution or different institution replicating the study, replicating the protocol. Classify this as no information if the study authors did not report whether the protocols were actually replicated by an independent group of researchers. Classify this as no or probably no if the study authors reported that the protocols could not be replicated by an independent group of researchers. So here they have actually tried and they could not replicate the experiments. Now, the next signaling question 5.3 is, did the authors or an independent group of researchers reproduce the results in similar and different laboratory conditions? Okay, how is it different from the previous signaling question? Okay, it's one thing to say that you could replicate the protocol, we could actually conduct the experiment using the same methods, but you may not get the results, similar results. So even when the protocols and methods are replicated by an independent group of researchers, the results may not be replicated or reproduced in similar or different lab conditions. So this signaling question assesses whether the results were replicated or reproduced. Previous signaling question was assessing whether the protocol could be replicated. Attempts to replicate or reproduce the results can be part of the same report, but it could also be another report, particularly if the attempt to replicate or reproduce the results is made by an independent group of researchers. So sometimes you can find it in the same report, sometimes from a different report. So the signaling question here, how would you classify it? Classify this as yes or probably yes if the results were replicated or reproduced. And as mentioned previously, it could be based on literature, it could be from the same study, it could be the independent group of researchers, could be from the same institution or a different institution. Classify this as no information if the authors did not report whether the results were actually replicated or reproduced by an independent group of researchers. So we don't have any information. Classify this as no or probably no if the study authors reported that an independent group of researchers could not replicate or reproduce the results.